This video will be talking about proteins. Proteins can be found in the phospholipid bilayer. Originally, in the Davson-Danielle model, the proteins were not embedded in the phospholipid bilayer. But they were wrong! But the Singer-Nicolson fluid mosaic model got it right by embedding the proteins in the phospholipid bilayer. This allowed solutes to transfer straight from one side of the bilayer to the other. There are three specific types of transport proteins: facilitated, passive, and active. Facilitated and passive transfer solutes from high to low concentration. This does not require energy. While active transport does the opposite. It goes from low to high and requires energy.
The function of proteins are support, transport, hormones, storage, defense, receptors, contractile, enzymes, and the most important are transport and enzymes. Enzymes alter other molecules in an orderly fashion by adding, subtracting, and changing shape.
Transport proteins are considered the gatekeepers of the membranes because they pump substances in and out.
The structure of a protein. A protein is made up of amino acids called monomers. When the monomers are connected by a covalent peptide bond, which creates a polypeptide, which is a polymer. One or more polypeptide chains makes a protein. There are 4 levels of structure for a protein. The primary structure is the sequence of amino acids. Hydrogen bonds between non-adjacent amino acids maintain the structure. The secondary structure is by Linus Pauling. This is the interaction among amino acids and the patterns of the hydrogen bonds. There's the helix on the left, which is slinky-like, and the pleated sheet on the right, which is fan-like. The hydrogen bonds allow it to stretch and move because they are variable in length. A tertiary structure is a larger scale interactions between the amino acids. A disulfide bridge occurs between very distant parts of the polypeptide chain, which keeps the structure. A quaternary structure is macro interactions, which is the whole protein. An example of this is the hemoglobin in blood.
Protein performance is determined by environmental conditions, the first being temperature, the second, pH, the third, concentration of substrate, and the fourth, presence or absence of oxygen. In extremes of pH or heat, this may denature a protein. This means that the hydrogen bonds are disrupted, the shape is lost, and function of the protein is also lost.